for target validation or in some early proof of concept studies. Each one of these programs has a different starting point. There are very particular entry criteria. There's different levels of support. And so in addition to taking a look at the funding opportunities that are available on the NIH website, I also encourage you to contact a program director. You don't necessarily have to worry about getting the right one the first time round. We all know each other, work with each other closely. But it's important to actually talk to a program director and find out from them which program is going to be best suited to your needs. And they can also provide you some guidance on how you might focus your research or focus your application to increase your chances of getting funded. I want to talk about two different programs that are being supported through this new center at NIH, NCATS, that supports translational research. And these are in the research service model. So for about 5 years or so now, NIH has supported the Molecular Libraries program. We give funds to a collection of centers around the country, screening centers that have different areas of specialization. And you can apply to work with these centers. They can help you adapt an assay that you have for a high throughput format. They can actually run the high throughput screen for you, screening your assay against libraries they have in-house. And they can also do some additional medicinal chemistry work to optimize any hits that come out of those screens. And of course, this is at no cost to you. So you're not getting funds for your own laboratory, but you're essentially getting the services of another laboratory that has expertise in this area. Jim and Glaze spoke earlier at this meeting about the NCGC, and that is one of the screening centers that's in this program. That picture is actually of their robots. NIH also offers resources to support IND enabling work. This program was formerly called NIH RAID. It's now evolved a bit. It's called Bridges now. NIH has a bunch of contracts with different CROs to support all the different kinds of work you would need to be able to prepare for an IND application to the FDA to be able to do clinical trials. And as was discussed earlier in this meeting, this work can be quite expensive and also requires specialized expertise. You often need GLP certified labs. So you can apply to get free access to these CROs that will do this work. And one of the pluses of going through this program, as opposed to setting up direct collaborations with the CRO, is that you don't have to go through the process of picking the CRO. Any of the companies that have been hired through this program have been through a very rigorous contracts review process and is also managed by NIH staff. So I want to spend a few minutes talking about a slightly different model for supporting drug discovery. This is a program NIH started about 1.5 years ago called the Blueprint Neurotherapeutics Network. And we started this program partly in response to our discovery that within our academic community, commonly medicinal chemistry was being done in a rather unrigorous or underpowered way. So I know the question came up yesterday whether 30 compounds, 30 analogs might be enough to get you to the point of having a preclinical candidate that's well optimized if you apply all the state-of-the-art informatics and computational tools. And we've actually funded within NIDS a fair number of projects in that vein where our academic groups have put one postdoc chemist on the project. They've maybe made a few dozen analogs primarily focused on potency, maybe starting to get into ADME issues. And the unfortunate reality is every single one of these projects has failed to meet its optimization milestones after a year. So sometimes you see they may have made a bit of progress improving potency, but they haven't been able to address their ADMET liabilities. And I think the reality is when you're trying to determine your structure-activity relationship around potency and perhaps multiple ADME liabilities, or maybe you have a solubility issue, you need the capacity to be able to generate a fair number of compounds in order to test your hypotheses. And that can be very, very expensive and require quite a bit of resourcing. As Bruce spoke earlier, Many of the collaborators with AMRI are putting 4 to 8 chemists on those types of lead optimization stages, and that can be really beyond what's available at a university. So NIH has responded to this challenge by setting up a virtual pharma network. So we've set up a series of contracts to do the standard stages of drug discovery. So we've got a chemistry contract with AMRI, we have a contract with Southern Research and with SRI International to do PK tox work. We have plans to set up additional contracts for later stage work, all the way through phase 1 trials. So you can think about these as being kind of each of the departments of a virtual pharma. And what we're looking for is researchers from academia and from small businesses to become the biology department. If you have a compound that might be a good starting point for drug discovery, and if you have a collection of bioactivity assays, in vitro and in vivo, that can be used to test analogs generated in a chemistry campaign, you can apply to join this network. And we will, if you're successful, we'll give you funding in the form of a small U01 reward to support your biology work. You'll also get access to all of these contracts. And we'll team you up with consultants who have had a very high-level experience running departments in the pharmaceutical companies and who are now independent consultants. We have consultants in medicinal chemistry, in assay optimization, in PK work, and we've set up a lead development team that will include you as the principal investigator, these consultants, also representatives from the contracts. For example, we have AMRI chemists working on these teams, and NIH staff. And this group meets every 2 weeks by teleconference, and you're essentially having a virtual lab meeting. This is where you map out the set of experiments that you'll either do within the CROs or within your own lab, and you get feedback from the consultants on the data that came in for the last round. So it's a highly collaborative model. And I do want to emphasize that you will also get full control over your IP. NIH has no interest in having any role in your IP. We really want to see you take the projects further on and fully develop them and commercialize them. We're just here as facilitators. So we currently have 7 projects in this network, and you may recognize some of these names. Marcy Glixman is one of our PIs. She has an ALS project with us. We currently have one Alzheimer's project from Steve Wagner at UC San Diego. It's quite a diverse portfolio. You'll note that most of these are university or academic labs, but we do have one small company represented, and we have a range of different disease indications here. This is open to any disease that affects the nervous system. So referring back to some discussions we've had earlier in this meeting about knowing mechanism of action, I think about 3 of these projects we're fairly confident about what the molecular target is, but the rest we don't. They were identified in phenotypic screens. And in fact, one of these, which is Ed Rubel's project, the lead came out of a zebrafish screen. So that's really one of our more adventurous projects here. All these projects really got going this fall in terms of starting to do optimization of their assays and doing some validation experiments, doing some ADME profiling, getting started looking at the chemistry. And these projects will be facing their first milestones in June of this year. So the sort of milestones we have in place are they have to have an assay that's suitably robust and reliable to support a chemistry effort. And there needs to be some evidence of an emerging SAR. You have to know that your compound is actually tractable for a chemistry effort. And we also are checking on replicating and validating some of the biology work. We do have one more receipt date for applications. The RFA should be coming out in the next few weeks. It's going through final stages of NIH clearance. And it will be posted on the Blueprint website. If you are interested in this program, come talk to me afterwards. I've got business cards with the website indicated on the back. The deadline will be in early June. And I encourage you to read the RFA very carefully to understand the entry criteria for what you need in terms of a background for rationale for this project, for the assays, and for the compounds. particularly when we're talking about what sort of biological basis of biology data you need. As we've discussed in the last day and a half here, it's not a simple matter of showing that your compound works in an animal model of disease, because there's so many caveats with these animal models. So there's much more there's some more guidance in the RFA about what we're really looking for. It's highly desirable, for example, to have some sort of molecular readout of activity that demonstrates that you're engaging the pathway you think you're engaging. Of course, the RFA will indicate what kind of criteria your assays need to meet. We'll also be looking for compounds that are good starting points for chemistry. So finally, I just want to remind you to please contact program staff anytime you have any questions. I've listed a few questions here to keep in mind. The more quickly you can get to the kind of core of what you're looking for, the more quickly NIH staff can direct you to the right type of program, make sure you're talking to the right people, and very quickly get you on your way to applying for appropriate support. So I'm happy to take any questions.
So, Diana, have we exceeded time quota? All right.
Just want to say thank you, everyone, for coming, and please have safe travels back. Hope you enjoyed the conference. And please remember to fill out your evaluation forms. Thank you.